Hello everyone, my name is Sumaya and welcome to a case study of using the Genomics2Proteins Portal, a resource and discovery platform for investigating genetic screening outputs to protein sequence and structure. Before starting the case study, a few info that might be helpful. If the portal is useful for your research, please cite our publication. We have a series of tutorials that are posted in the G2P Portal's YouTube channel. If you don't want to miss a new case study, then please go ahead and subscribe. Finally, do you have feedback for us? Want to partner with us for a specific analysis or collaboration? Then reach out to us via the feedback form on the homepage of the portal. So with that, let's start today's case study, which will focus on analyzing the protein or molecular level effect of a clinically identified variant in KIF2A with significance level listed as uncertain significance in ClinVar database, which means that with the current set of evidence, the variant cannot be classified as benign or pathogenic. But what's the effect of this mutation on protein? That's what we will try to answer in this use case. A little spoiler, we will also see that this particular variant is annotated on a non-canonical transcript, and we will see how you can use the Genomics2Proteins Portal to analyze the protein effect of a non-canonical transcript variant by mapping it onto the canonical protein. All right, so let's go to the portal, and we will use resource module for this case study. And now we will type the gene name to load the gene page. And here, the first tab that is open is the protein sequence annotation with the protein feature viewer, where you have a suite of protein sequence, structural, and interaction, and also functional features that you can display at the amino acid level. And then we will go to variant to protein sequence tab to find the target variant of interest for this particular tutorial. So after you load this tab, you will see that by default, we have a protein isoform selected, which is a non-canonical protein isoform. So we will first select the canonical protein isoform per UniProt database, which is marked with this asterisk here. And then we will scroll down to identify ClinVar variant. However, the portal will display now that all ClinVar variant for this particular gene are annotated on a different transcript, which is not the transcript that is associated with the canonical protein isoform. So we will use this particular feature offered by the portal, and we will align variant from another transcript. So we'll click here, and we will identify this particular transcript that ends with 1,8, which is this one. This is the canonical transcript, but not matched with the canonical protein isoform. So we'll select this one, and now you will see that ClinVar variants are mapped onto the protein sequence. But by default, we show only the pathogenic variants. But remember, this particular target variant of interest is a variant of uncertain significance. So we will click here to bring in all the VUSs and map them onto the protein. So our variant was this particular glycine to arginine mutation that is located onto the 478 position of the protein sequence. So we will zoom in and we will identify that position, which is this one. And now on the platform, you can click on this orange sphere to open the details of this variant and also the protein feature of the reference amino acid as the card information. So in this variant information card, you can identify this particular mutation that we are currently analyzing, listed as a VUS along with the phenotype information. And now let's look at the protein feature card to understand the protein level effect of this particular mutation, glycine to arginine. So at the amino acid level, the impact is substantial because the glycine and arginine are two very different amino acids. And also, according to the structural feature, according to multiple structural features, this particular mutation is located on a helical structure, pretty much in the core of the protein, and also has a very high confidence level per AlphaFold, which demonstrate that this particular mutation will have an effect on structural features of the protein. And additionally, this mutation seems to impact the protein-protein interaction as well, according to this experimentally solved structure 6BBN. So it breaks a hydrogen bond. And also, this particular mutation is located on the protein domain as well. So because we understood now the protein structure that have an effect by this mutation on the interaction site, let's try to investigate this protein structure and find out where exactly this mutation is on the structure. So for that, we will go to the next tab, variant to protein structure, and we will click on this experimental structure 6BBN. So after the page loads, we will do the similar exercise to map the non-canonical variant to canonical protein isoform first. So we'll use the align variant feature, and we will bring in this particular transcript, and we will select all the VUSs to bring our target variant onto the protein sequence. So I believe this is the particular variant at the position 478. And now let's look at the structure. So this colored structure is the structure of the protein encoded by this gene. Now to see the mutation on this structure, what we will do is we will click on this row header on the sequence viewer, and that will map all the mutation as orange spheres on the structure. So now when you hover over the mutation position on the sequence, it will actually show that where this mutation is on the structure. Now it highlights it, and it will also display the disease annotations and protein consequences from the ClinVar. So now with that, we now know that this is the position of the mutation, this particular sphere. Now let's also map a protein feature onto the structure. So for that, we will hit this domain, and that will highlight the part of the protein that is that particular domain. But our target variant of interest is this particular position. And by mapping it on the structure now, we can clearly see that this particular variant is at the protein-protein interface interacting with this particular protein, and it is on a helical structure, and it is very likely that it will have an impact on protein structure and function. So I hope this tutorial has been useful. Come back again for a new tutorial. Thank you.